In this video, we will discuss about this interleukin 12 based nanoparticles interactions with the HM1 cells, and we will study these interactions in the in vivo studies so that we can characterize the function of these IL-12 based nanoparticles that they have developed. So in the panel A, they shows, so before going into the results, let us just quickly understand what they did the experiment. So they took the mice and then inject them with these HM1 cells, which is similar to the tumor cells. And after one day, they injected these IL-12 based nanoparticles, and for the control they also injected the traditional nanoparticles that does not have this IL-12 and the other polymer layers. And thereafter, after 14 days, they analyze the tissues, and in between they also analyze the interleukin 12 nanoparticles expressions. And in this study, they have used HM1 cells, which is Luciferase, which has a Luciferase reporter, and they use nanoparticles, which has a fluorescence, and also they have the interleukin 12 fluorescence. First, what they did is so let's now go into the results. So in the panel A, the results shows that for this unlayered or traditional nanoparticles, they are able to see, for this traditional based nanoparticles, they are able to see high amount of this nanoparticle based luciferase compared to the case where they used for the traditional nanoparticles, that is unlayered nanoparticles, the amount of luciferase signals is low, indicating that these unlayered nanoparticles degraded at a faster rate compared to the case where they use layer by layer nanoparticles that has the interleukin 12 and other polymers. Thereafter, they check the interleukin 12 signals. So in the case when they use unlayered nanoparticles, they are able to see interleukin 12 is retention times is also lower, and when they use the layer by layer based nanoparticles, they are able to see its retention time is higher compared to the case where they use unlayered nanoparticles. So far, the results from the interleukin 12 based nanoparticles seems to be better, as they have the high lifetime, or they are able to retain in the system for a longer period of time. At the same time, interleukin 12 is also retained in the system for a longer period of time in the case where they use layer by layer based nanoparticles that have this interleukin 12. So now let's let's see the results of the tumors. So in terms of tumors, we can see that the unlayered, in the case where the unlayered nanoparticle is used, the peak rise in the unlayered based nanoparticles is high compared to the case where they use layer by layer nanoparticles. Let me correct this. So this layer by layer nanoparticle signals are red. So the results for the layer by layer nanoparticles shows a high peak. However, the final results after few hours, this seems to be same for the layer by layer nanoparticles and for the unlayered nanoparticles, and they check it at multiple organ sites, and the significant changes that they have observed is for this UGT and omemtum, and the results for the other organ sites are shown in the panel F, where we can see that the results are non-significant except for the case this UGT and omemtum. And here what they have seen is in terms of the results is that when they use this layer by layer nanoparticles, they are able to see high amount of this nanoparticles presence for both UGT and omemtum compared to the case when they use unlayered nanoparticles. So this further indicates that the efficacy of this layer by layer nanoparticles is higher compared to the unlayered nanoparticles. Thereafter, they also checked the correlations between the HM1 cells, which has the Luciferase reporter, and this interleukin 12 cells, or this LBL, layer by layer nanoparticles, which also has a fluorescence reporter. And they are able to see for the layer by layer based nanoparticles, the correlations coefficients are higher compared to the case where they use unlayered nanoparticles. And one of the representative image is shown in this panel H, where we can see that for the unlayered based nanoparticles, the this the overlap is not 100%, or we can see that the presence of these unlayered based nanoparticles is not at all the sites, whereas when we see it for the layer by layer based nanoparticles, the overlap seems to be almost close to 100% in this representative image. This indicates that the high specificity of this layer by layer based nanoparticles compared to the unlayered nanoparticles. So that's all for this video. I hope you understood how they characterize their IL-12 nanoparticles in the in vivo studies with this HM1 cells. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.